Previously, we defined homeostasis as a state of steady internal conditions that is maintained by all living things. And maintaining homeostasis requires that the organism continuously monitor its own internal conditions. Now, for humans, we have organ systems such as the nervous system and endocrine system, which are sensitive to any changes that occur internally, and then one of their primary functions is to then communicate those changes with other organs and other organ systems so that those organ systems can alter what they're doing in response to that change and bring the body back into homeostasis. So for each physiological variable or value, that value has a set point about which a normal range of fluctuation will occur. And we'll see an example of this with body temperature. I'll show a few graphs here to illustrate that point as well in just a moment. But basically what we see with that type of homeostasis is that when deviations from that set point occur, the body will use a mechanism called negative feedback in order to bring that particular value or variable back to the set point and within that normal range. So if we very simply graph this out, homeostasis being a narrow range around a set point, we can graph our variable of interest on a vertical axis here and time on our horizontal axis here. Our set point for our particular variable here will be our purple dashed line, and the narrow range of fluctuation about which that variable can fluctuate or deviate is highlighted by this, this blue shaded region here. So if this, for example, were body temperature, our set point for body temperature, typically about 37 degrees Celsius, about 98.6 degrees Fahrenheit. But if you were to measure your body temperature at any given moment during the day, or even throughout the day, you would find that some points during the day, your body temperature might be a little bit higher, 98.6. Other times it might be a little bit lower. And that's okay according to homeostasis, that there is a little bit of fluctuation around that variable, but as long as our body temperature doesn't get too far beyond that set point, then the body considers that variable to be in our normal typical range for temperature homeostasis. We can see a man-made example of this by looking at how an air conditioner system works in our homes. So we've got temperature here in this degrees Celsius on the vertical axis and time on the horizontal axis here. We have our set point, which in this case is 22 degrees Celsius, which is about 72 degrees Fahrenheit, and the normal range of fluctuation of that particular variable there. So at any point during the day, let's say that in the morning when the sun rises, starts to heat up our home, temperature starts to rise, at which point the air conditioner will turn on after the temperature in our house has exceeded that 22 degrees Celsius, 72 degrees Fahrenheit set point. The air conditioner will turn on, start to slow the increase in temperature. So it will eventually start to bring the temperature below that set point, at which the air conditioner will turn off, and the house will continue to cool for a little bit. So as long as our temperature fluctuates about that normal range, then everything is considered to be in homeostasis with that particular system. What we just saw in those examples with body temperature and with the example with the air conditioning are examples of negative feedback mechanisms. We'll spend a little bit of time on this because negative feedback has a couple of basic components that we'll examine. The first being the stimulus, which the stimulus is simply the change in the physiological value from the set level of homeostasis. So in that case, our stimulus was an increase in body temperature or a decrease in body temperature, an increase in room temperature, a decrease in room temperature. That would be the stimulus for our negative feedback mechanism. We need to be able to detect those changes when they occur, and those are the roles of the sensory receptors, the various sensory receptors located throughout the body that are sensitive to different stimuli. So in general, a sensory receptor is a structure that monitors a specific physiological value for a change from homeostasis. We also have the control center, which is a system that's going to receive information from the sensory receptor, and it's going to compare that information to the normal value or normal physiological range for homeostasis. We also have an effector, which is a structure that the control center will activate if that physiological value is too far beyond the set point. So if it's either too high or too low from that set point, then the effector is the structure that's going to be activated in order to move that physiological value closer to the set point. And finally, we have the response, which is what the effector does in order to change that physiological value of interest. And the response is really the activation of those structures to return that physiological value back to homeostasis. And the reason why it's called negative feedback is because the body's response to that particular change and the initial stimulus are in the opposite direction of one another. So for example, when our body temperature rises, becomes too warm, the stimulus of our body temperature rising, the response of the body is to sweat and bring blood flow closer to the surface of the skin. Both of those responses are going to have the effect of reducing body temperature, getting rid of some of that excess body heat. So in that way, the response and the initial stimulus are in the opposite directions of one another. I discussed this one very heavily just because negative feedback mechanisms are the most common feedback mechanisms in the maintenance of homeostasis. So examining, once again, kind of a very simple diagram. This is figure 1.10 of negative feedback mechanisms from the textbook. We see that there is an initial stimulus, which will activate a sensory receptor or will be detected by a sensory receptor. That sensory receptor will then communicate that change in the physiological variable to the control center. The control center will then activate an effector, and the effector will create a response to that initial stimulus. And that that final response will feed back to reduce the effect of the initial stimulus. So once again, with our example of body temperature here, we see that when body temperature exceeds 98.6 degrees Fahrenheit, 37 degrees Celsius, we see that there are nerve cells that are activated in both the skin and the brain, which tells our brain that body temperature is rising beyond what it physiologically should be. There's a temperature regulatory center in the brain, which will then activate sweat glands located throughout the body. Sweat will be produced, and then that will increase the heat lost from the surface of the body. So that way, that response will feed back to reduce the increase in body temperature that was the initial stimulus. We examined negative feedback mechanisms because they are the most common feedback mechanism in homeostasis regulation, but we also have positive feedback mechanisms, which the function of positive feedback mechanisms is to intensify a change in a physiological value rather than reversing it, because we saw that negative feedback mechanisms function to reverse changes in physiological values, whereas positive feedback mechanisms, not quite as common, but they still do serve a very important purpose in the body. So positive feedback is a normal feedback mechanism only when there is a definite endpoint to that particular activity or that particular thing that's going on within the body. And we've got kind of two primary examples of this. The first is contractions of the uterus during childbirth. The second is blood clotting. So we'll examine the childbirth one in just a moment. But what we really see with contractions of the uterus during childbirth is that the head of the developing fetus is going to push against a region in the uterus called the cervix. The cervix is going to communicate that information to the brain. The brain is then going to stimulate a gland called the pituitary gland to secrete a hormone called oxytocin. Oxytocin will then stimulate contractions of the uterus, which further kind of pushes the head of the fetus, the baby, against the cervix and kind of promotes uterine contractions that are seen during childbirth. And the second is blood clotting, in which we see that there are chemical factors that are released when blood vessels are damaged. And those chemical factors will stimulate the gathering of these small cell fragments called platelets at the site of the damage. Platelets, once they are activated and at that site of damage, they will release chemical factors that recruit additional platelets to that site, and that feedback mechanism will continue until there's enough platelets gathered at the site of damage to essentially block or plug up that damaged blood vessel with a platelet plug. So we'll examine in this last diagram here for this particular section positive feedback mechanisms in childbirth. So we see that as the baby gets larger in size toward the end of normal pregnancy, the head of the baby is going to push against the region of the uterus called the cervix. Nerve impulses from the cervix are going to be transmitted to the brain. The brain will receive that information and communicate with the pituitary gland to secrete a hormone called oxytocin. Oxytocin is released from the pituitary gland into the bloodstream. That oxytocin will be carried to the uterus, where it will then stimulate the contractions of the uterus, which is going to amplify the pushing of the baby's head into the cervix. And eventually those contractions will become so frequent, so strong, that childbirth will occur.